Okay, I think I'm live, but give me a second to find out. Okay, I'm going to start going through some of these questions that I got through the survey submission. Let me screen share. Okay, so the first question is, give me one sec. Okay, can you explain the phases of mitosis and meiosis? I'm going to hold off on doing that just because there's so many good videos. Let me get to some of the other questions. What is CAMP and AMP and cyclic GMP and GMP, and how is it related to transduction pathways? Okay, so let's talk about secondary messengers. So this is ATP. You have a sugar ring and a nitrogen-containing ring of carbon, hydrogen, and nitrogen atoms. And we call this ring adenine. And then if I'm drawing ATP, it would be 3-phosphate groups. So AMP is just one phosphate group, and cyclic AMP is, so this is AMP, and cyclic AMP is when I form a covalent bond between this phosphate and this carbon right here. And cyclic GMP looks very, very similar. The nitrogen-containing ring is slightly different, so we call it cyclic GMP instead of cyclic AMP. But the whole purpose of secondary messengers is that these proteins are going to bind to kinase enzymes, and by binding to kinase enzymes, they're going to activate them so that these kinase enzymes can go off and phosphorylate other proteins and get that phosphorylation cascade going. There are enzymes called phosphodiesterases, and these enzymes will hydrolyze this covalent bond in order to destroy the cyclic nature of the compound. And destroying that prevents these molecules from activating kinases. So that's how that secondary messenger concentration is regulated in the cell. So to answer your question, how is it related to transduction pathways? They help initiate the phosphorylation cascade. Chapter 43 was 22 pages long, but how in-depth do we need to know about it? You need to know all of it. Can you explain the role of the checkpoints in the cell cycle? The role of the checkpoints. The role of the checkpoints is just to ensure that there are no fatal errors or major errors in the cell cycle, and for most of your cells, the checkpoints are successful in catching and fixing any errors. Sometimes, though, that process messes up. So what I'm drawing right here is cytokinesis, which is the last part of M phase. This purple is mitosis. And we can break this up into G1, S, and G2. So there are probably a lot more than just 3 checkpoints, but the major checkpoints are at the end of G1 to make sure that the cell is ready to invest the energy necessary to copy its entire genetic library, and then right before it starts doing mitosis, and then right before it splits off. So again, these checkpoints are just one mechanism that tries to minimize any errors in mitosis and also any errors during DNA copying, because we don't want the daughter cells to have mutated DNA. So if one of the checkpoints does fail, there are proteins that will put everything on hold. It will stop the cell, and it will give the cell, it will activate proteins that will try to fix any errors that are detected. And if the cell successfully corrects those errors, it goes back into the cell cycle. If it fails to correct those errors, the cell is instructed to commit apoptosis. All right, since I don't see any more questions, I can go back to the mitosis and meiosis question. We can break this down into mitosis and meiosis. So this is broken up into prophase, metaphase, anaphase, telophase. And this is broken up into meiosis 1, which itself consists of prophase 1, metaphase 1, anaphase 1, telophase 1, and then cytokinesis. And then meiosis 2, which consists of prophase 2, metaphase 2, anaphase, telophase 2, and cytokinesis. So some of the big differences here is that in prophase 1 of meiosis 1, you have crossing over. You do not have that. You do not have crossing over in mitosis. The whole point of crossing over is to create genetically unique daughter cells. That is not the function of mitosis. It's not trying to create genetically unique daughter cells. Another big difference is that during metaphase 1 of meiosis 1, the homologous pairs will line up next to each other. So this is the chromosome that you inherited from mom. This is the chromosome that you inherited from dad. That's how they line up during metaphase 1 of meiosis 1. In mitosis, they would line up something like this. Okay. So because they're lining up like this over here during anaphase 1, they are actually going to, hang on. The homologous pairs are going to separate. The sister chromatids are not going to be pulled apart during anaphase 1 of meiosis 1. Over here, the sister chromatids are pulled apart. And then telophase 1 of meiosis is very similar to telophase of mitosis. And everything about meiosis 2 is pretty much the same when you compare it to mitosis. So meiosis 2 is virtually identical to mitosis in terms of how the chromosomes orient themselves and what happens during the PMAT. There's a question, during which division is the chromosome number reduced? That's during meiosis. The chromosome number, I think what they're referring to is the diploid number. The diploid number after meiosis 1 is reduced to 2 haploid cells. And when you split those chromatids apart, you still end up with 4 haploid cells. But the reduction aspect of cell division happens at the end of meiosis 1. Are haploids and gametes synonymous? Kind of. I mean, I think the chapter uses them synonymously, but basically I could refer to these cells as haploid cells, but I technically couldn't refer to them as gametes because by definition, gametes are mature sex cells, like they're sex cells that are ready to go. So these cells are haploid, but they're not fully mature. They're not ready to fertilize, so I really wouldn't refer to them as gametes. I'm seeing a lot of stuff about chapter 3. Okay, or chapter 43, I mean. So I'll quickly go over the immune system. So the immune system is, I guess we can start with, let's just start with the inflammatory response. All right, so during the inflammatory response, you have something is going wrong. Your innate immune system has been alerted, it's been activated, and some of the things that are going to happen are, let's just say that you have bacteria that's been exposed to your tissue. They have broken through the barrier defenses like the skin, the mucus, the secretions, and so you have white blood cells that are just hanging out, waiting for the opportunity to do their job. And a white blood cell like a phagocyte, that could be a white blood cell, or a macrophage or a neutrophil, is going to gobble up. It's going to do phagocytosis. It's going to eat up the pathogen that's entered your tissue. And in the process of doing that, it's going to secrete signal molecules. Your book calls them cytokines. So these and other signaling molecules like histamine might be secreted by mast cells. But basically, these signaling molecules diffuse into the tissue, they diffuse into the circulatory system, and they recruit your white blood cells. They recruit white blood cells like eosinophils, basophils, all of your granulocytes, your phagocytic cells, your dendritic cells. These are sort of like the first responders. They're going to invade the area that is that's in distress, and they're just going to start gobbling stuff up. And as they gobble stuff up, well, let me go back to here. So when we're recruiting white blood cells, that's what causes the symptoms of inflammation, like redness, because your circulatory system becomes leaky, which is going to allow even more white blood cells to easily squeeze out of your circulatory system and into the area that's in trouble. Heat, because you have warm blood that is coming from the core of your body leaking out into your, your finger. Let's pretend that you got a splinter. Some other symptoms include pain. So as the fluid builds up in your tissue space, your interstitial space, that swelling is going to press on nerve endings. And also some of the molecules that are secreted by your immune system is going to cause pain neurons to depolarize. So a combination of these, the swelling and the molecules that are secreted by the immune system, causes pain. Am I missing something? Probably. But these are some of the major symptoms of the inflammatory response. And getting back to recruiting the white blood cells. So these white blood cells are going to gobble up the pathogen. Let's call this a dendritic cell, and it's considered to be an antigen-presenting cell because what it'll do is it'll do phagocytosis. So these are little phagosomes that have the little pathogen in it. And give me 1 second, somebody's mic is on. and turn your camera off. Okay. This pathogen is going to get broken down. Lysosomes are going to fuse to it. It's going to get digested by hydrolytic enzymes. And then the dendritic cell isn't done. It's going to display its trophies. It's going to take fragments of what it destroyed, and it's going to present them on plasma membrane proteins that we call MHC2. And so it's presenting antigens, it's presenting fragments of destroyed pathogen. And this dendritic cell is going to travel through the lymph system and hit your lymph nodes. And inside the lymph nodes, there are lymphocyte cells, and that includes helper T cells, immature helper T cells. I'll write an I for immature. It includes B cells, immature B cells, and also immature cytotoxic T cells. A lot of other things, but those are the cells that we focused on. Give me 1 second. Okay, so these helper T cells have created, by randomly cutting up their DNA, these helper T cells have created receptors that have a variable region that may or may not recognize the antigens that are being presented by this antigen presenting cell. So let's pretend that this helper T cell happens to recognize the antigens that are presented by the helper T cell right here. So what's going to happen is this dendritic cell is going to, with its MHC2 and its little pathogen antigen fragment sticking out, it's going to bind to the T helper cell receptor. That T helper cell has an accessory protein called CD4 that's going to sort of grab on to MHC2, and these molecules, or I'm sorry, these cells are going to start talking to each other. The antigen presenting cell is going to talk to the T helper cell, and the T helper cell is going to talk to the antigen presenting cell. And at the end of that communication exchange, that T helper cell is going to graduate from an immature T helper cell. It's going to turn on and become a mature T helper cell. And it's going to divide, and it's going to turn into an effector cell and a memory cell. Now, the memory cell is just going to hang out and wait for years or maybe even decades to be activated again. It's going to immediately, if it ever encounters this antigen again, it's going to immediately turn on. Hang on, I did something wrong here. The effector cells, though, are going to go look for other lymphocytes to activate. So the other lymphocytes that they can activate are the B cells and the immature cytotoxic T cells. So let's talk about the B cell. The B cell, while all of this is happening, there are B cells that have receptors. And these receptors are basically just membrane-bound antibodies. They will shuffle their DNA just like a helper T cell, and this region of the antibody that I've just drawn in a box differs between every single B cell. So one B cell might look like this, another B cell, because they shuffled up their DNA slightly differently, might have a antibody receptor that looks like this, and so on and so forth. And so this activated helper T cell, one that I was just talking about right here, is going to go and look for a B cell that is displaying that exact same antigen. So let's say that that antigen bumps into this B cell right here, but this B cell doesn't have the right receptor. The receptor and the antigen don't interact strong enough. So it floats around and it eventually bumps into this receptor. And this receptor happens to create a very strong interaction with the antigen. And so the B cell is going to, not through phagocytosis, but through receptor-mediated endocytosis, is going to take that antigen in, and just like over here, it's going to display that antigen on its MHC2. Okay? So this T cell over here that was activated and turned into an effector, mature T helper cell, this T cell is going to, I'm running out of room, is going to bind, just like it did with the other antigen presenting cell, it's going to bind to the MHC2 and antigen complex. And with its CD4, it's going to latch on to the MHC class II molecule. And just like over here on the left, the B cell and the T cell are going to start talking to each other through paracrine signaling and autocrine signaling. And eventually what's going to happen is this B cell is going to get turned on, and it is going to graduate into a mature B cell, just like what happened over here with the T cell. And this B cell is going to divide, and it's going to turn into an effector cell that we call a plasma cell. And it's going to also, one of the daughter cells is going to remain a memory cell and just wait for exposure to that antigen again. This plasma cell is basically going to, all of this antibody right here, this receptor antibody, it's basically going to do massive protein synthesis of that antibody. And it is just going to crank out through massive rounds of exocytosis. I think it's something like 2,000 of these proteins per second. It's going to crank them out. And these antibodies are going to go and bind to the antigen. So if this is like the little pathogen with its little, I guess, I guess I drew the antigen as like little purple things sticking out. It's going to go bind to these antigens, and it's going to flag it for destruction. We call that opsonization. Or these antibodies can also recruit complement proteins. Remember, complement proteins are proteins that are like little daggers or little drills that just drill or poke holes through the plasma membrane of the pathogen. And it's also gonna sort of like create a shell. These antibodies kind of create a quarantine shell around the pathogen, right? And what makes this so effective is that this is just one single antigen. This whole thing that I've just talked about is one single antigen. This pathogen can have thousands of antigens on it. So I could draw thousands of different proteins, or I'm sorry, thousands of different antibodies that were activated that target, each one of them targets a different region or a different antigen on this pathogen. And then the exact same process happens with the cytotoxic T cell. The cytotoxic T cell can be activated by T helper cells, and once it's activated, it's going to mature, it's going to divide and turn into an effector cell and a memory cell, and it's not going to secrete any antibodies. What it's going to go do is it's going to go look for cells that are infected. So if a cell gets infected, or if a pathogen finds its way into a cell, the cell is not going to go down without a fight. And the way that the cell makes its last stand, kind of, is it will take parts of the pathogen that entered it, and it will display those parts on a protein called MHC1, not 2, but 1. And if an activated cytotoxic T cell recognizes this antigen that the cell in distress is displaying, it will basically secrete molecules that ultimately induce apoptosis. So this cell is sort of saying, it's better for me to die than to turn into a pathogen factory. Right? So we're picking off pathogens, we're preventing pathogens from thriving in the body. We are recruiting the immune system through the inflammatory response. This is one big, massive positive feedback system. That was a long time, so let me see if there are other questions. Does the same process apply to an infected cell and MHC1 cytotoxic T cell? Yes. What do replicated chromosomes look like? They look like this. This is chromosome number 1 that you got from mom. This is chromosome number 1 that you got from dad. Now, during S phase, these chromosomes are duplicated, but chromosomes really don't exist during S phase. They only exist during mitosis. But anyway, the DNA is replicated so that if we could see the chromosomes during S phase, the chromosomes would look like this. So this, what I'm drawing right now, is one side is referred to as a chromatid. This is a chromatid, this is a chromatid, and I refer to this structure as a duplicated chromosome, and this could be called a non-duplicated or an unduplicated chromosome. And so the same thing would happen over here. You would have, remember, these chromatids are letter for letter identical in sequence with the exception of mutations. And so that way, when the cell splits thanks to mitosis, this one will go to that daughter cell, this one will go to that daughter cell, and this chromatid will go to that daughter cell, and this chromatid will go to that daughter cell. And so that way, each daughter cell still is a diploid. This daughter cell is a diploid, and this daughter cell over here is a diploid. Okay. All right, I'll wait for, I don't see any more questions, so I'll wait for another question. Does the cell produce diploids through mitosis? Yes, the daughter cells in us, the somatic cells, when they divide by mitosis, each daughter cell is also a diploid. Otherwise, every time a cell divides, it loses genetic information. Well, we'll talk about that later. But yes. While you wait for other questions, can you hit on some other main points in chapter 43? Can you explain the whole HIV process again? Let me do HIV, and then while I'm doing that, I can try to hit up on some other points of chapter 43. So you really just need to know some of the basics about HIV, and some of the basics are it's a retrovirus, which means it has RNA as its genetic information. On the outside is plasma membrane, or the phospholipid bilayer that it steals from its host, and its host is helper T cells. So there's that phospholipid bilayer, and then it's got like a capsid and then another protein layer called a matrix, but we'll just focus on the plasma membrane, or the phospholipid bilayer, and then the capsid on the inside. There's some RNA on the inside, and there's also some enzymes, some proteins that help the virus successfully invade and then get out of the host cell. The only protein, well, GP120 is one of the proteins that we talked about, and it's important because it is used to invade a helper T cell. And then we talked about a protein called P24, and this is just what Paxton measured in lymphocytes to determine whether or not these lymphocytes were able to resist infection. It's just a capsid protein. So if, depending on what strain we're talking about, and I don't even know if I remember the difference. I know that there's M and T. I think M is CCR5 and I think T is CXCR4, but I'm not positive. Either way, these strains rely on accessory proteins on the T helper cell in order to infect the T helper cell. So we're looking at a T helper cell, not drawn to scale. This virus is much, much, much smaller. And the virus is going to use CD4. Remember, CD4 is used by the T helper cell to grab on to other cells and begin communicating with them. And then depending on the strain will either use CCR5 if it is M-tropic, or CXCR4 if it is T-tropic. And I need to check that because I don't want to tell you the wrong thing. So give me 1 second. Okay, that was right. So let's see. So what did Paxton do? Paxton basically, he determined that it was the T helper cells that were making individuals resistant to HIV, and he took these two different strains of HIV and he exposed them to lymphocytes from people who were immune to infection, or at least resistant to infection. Let's say resistant to infection. So these people had T helper cells with CD4, and each T helper cell also has CCR5 and CXCR4. And he saw that when he infected these, or when he exposed these lymphocytes to the M strain of HIV, HIV could not infect these cells. However, when he exposed these same cells to the T-tropic strain of HIV, they were able to infect the T helper cells. So this told Paxton that basically these individuals were resistant because they have an altered CCR5 protein. It's either altered or it's missing altogether. And so since this protein is altered, let's just say it's altered and it has like a weird shape. Since it has a weird shape, HIV can't associate with this protein. And since it can't associate with this protein, it can't successfully infect these lymphocytes. So what else about HIV? If it is able to infect a lymphocyte, like a T helper cell, it will take its RNA. I'll draw it in blue. It'll take its RNA and it will convert it to DNA using an enzyme called reverse transcriptase. So I'll draw the DNA in red. So DNA is double-stranded, so it creates two DNA or two nucleic acid DNA strands. And the process of doing this is prone to error. This is actually advantageous for HIV because it means that mutations crop up pretty often, which means that HIV is constantly changing its antigens, which makes it very difficult for memory cells to ever figure out or mount an effective defense against this pathogen because it's constantly changing the way that it looks. It uses some other enzymes to actually take its DNA and stitch it into the host genome. We call that integrase. And then finally, it uses still other enzymes that it packages inside the capsid to assemble the capsid and bud off of the host cell so that it can go and infect other helper T cells. And so what makes HIV so deadly is that this process that I described up here, basically it gets shut down because HIV is destroying these cells, which means your body is unable to activate B cells and unable to activate cytotoxic T cells. So your immune system basically crashes. No bueno. No, you do not have to know the special antibody classes. Yes, I can go over muscle contraction. Let me see what else. What exactly are IP3 and DAG used for? Those are in chapter 11. They're just other examples of secondary messengers, but other than the fact that they're secondary messengers, you really don't have to know specifically what they do. They just help relay a cell signal. Muscle contraction, okay. I can do that. Muscle contraction, we are going to look at a muscle fiber cell, which is a really, really, really long cell that is filled with protein filaments called myofibrils. So let's zoom in, and your textbook has, or just even Google Images has so much better pictures than this. So if you need to pull that up while I'm talking about it, feel free to. Let's zoom in on one myofibril. If we were to zoom in on a myofibril, we would see something that kind of looks like this. It's sort of like little bricks, or little rectangles next to each other. And one end to the next end, so one little brick is what we call a sarcomere, and it's the fundamental contractile unit of a muscle cell. And the anatomy of a sarcomere is you've got proteins called actin filaments that reach towards the middle from each side, and in the middle, reaching out towards the sides, you have myosin. And during a contraction or during a power stroke or during the sliding filaments, what you have is you have the brick, this rectangle sort of crunches in, it gets pulled inwards. And the reason for that is because these blue myosin heads are literally pulling on the actin filaments. And as they pull on the actin filaments move towards each other. And again, I've posted videos that go over this in detail. So, you know, if you want a little bit prettier of a picture, please watch those videos. But also remember that this actin has another protein called tropomyosin wrapped around it. And so myosin can't pull on actin unless tropomyosin gets out of the way. And I should have drawn this bigger, so let me do that real quick. Let me just draw one sarcomere. In order for tropomyosin to get out of the way, we need calcium because there's a fourth protein. So there's the actin, there's the myosin, and I'm going to stop there. Let's just get to the tropomyosin up here. There's a fourth protein called troponin. Troponin sort of like sits on top of tropomyosin, and it is going to bind to calcium. When calcium is present inside of the cell, inside of the cytoplasm, that calcium will bind to troponin. Troponin will shift, which will cause tropomyosin to shift, so it gets out of the way. And now the myosin head is free to do the power stroke. So the power stroke is just the sequence of ATP hydrolysis and ADP and inorganic phosphate displacement that results in a head reaching up, coming back down, letting go. Reaching up, coming back down, letting go. That's the power stroke. And so all of that is happening when a muscle fiber, I'll go back up here. When a muscle fiber is depolarized thanks to a motor neuron. So a motor neuron, the axon terminal of a motor neuron is going to depolarize. That's going to cause the motor neuron to spit out acetylcholine. Acetylcholine is a ligand that binds to ligand-gated ion channels called, I guess they're just called acetylcholine receptors. That's going to cause the receptors to swing open and sodium to flood in. And when sodium floods in, it's going to cause the muscle fiber to depolarize. And when the muscle fiber cell depolarizes, calcium diffuses out of the ER and is able to bind to troponin. What is an epitope? An epitope is just a specific region of an antigen. So going back up here, I spent a lot of time talking about this one antigen that activates this one helper T cell. Well, in reality, if we were to let's zoom in on this antigen. If I were to zoom in on this antigen really kind of looks like this. All right, that is what the antigen looks like. So in my example up here, maybe the T cell that I was talking about binded to this region of the antigen, but that's just one region that could elicit an immune response, or one region that could result in the clonal selection of B cells. This region could activate other helper T cells and other B cells. This region can activate other helper T cells and B cells. This region could activate a different set of T cells. So you get the point, is that one single antigen can activate hundreds of different B cells, which will secrete their own antibodies that target that one specific region of the antigen. So that's what an epitope is. Does Viagra use cyclic GMP as a secondary messenger? No, cyclic GMP is a secondary messenger that results in smooth muscle relaxation. Viagra's job, or its mechanism of action, is just to prevent cyclic GMP from being broken down. Not completely, but just slow down the rate at which it's broken down. So when guanylyl cyclase is activated, typically by the signaling molecule nitric oxide, this guanylyl cyclase will take GTP, which looks something like this, and it will hydrolyze GTP right here, break off those two phosphate groups, and convert it into cyclic GMP. That's what guanylyl cyclase does. Now, this is a secondary messenger that will go and activate a whole bunch of proteins that ultimately result in smooth muscle relaxation. Now remember, there are enzymes called phosphodiesterases that will hydrolyze this bond and shut down the signal. Viagra's job is just to prevent those enzymes from breaking down cyclic GMP. So here's phosphodiesterase. It normally attacks this covalent bond. Viagra gets in the way. So it's an inhibitor. I don't see any more questions, so I'll wait. Are local regulators mainly used to signal cell growth? Those are the only local regulators we talked about. Yes. So those would be like growth factors that bind to receptor tyrosine kinases and induce mitosis in neighboring cells. What is crossing over in meiosis? Crossing over is just where, okay, so this is considered, these are considered to be sister chromatids. This is a homolog, and the red sausages are sister chromatids. Crossing over is exchanging material, exchanging pieces of chromosome between homologs. Another way of saying that, it's exchanging material between non-sister chromatids. So I might see something where this chromatid leaves and goes over here, or I'm sorry, this part of the chromatid leaves and goes to its homolog. So it'll travel over here. Oh no, what did I do? Let's try that again. And the swap is always going to be equal unless something goes wrong. But assuming nothing goes wrong, the swap is always equal, right? So what at the end of it all, what we might see is, we might see a chromatid that kind of looks like this. And the whole point of this is just to create a chromatid that has different combinations of alleles compared to what it used to have. So this is just creating diversity. It's creating a genetic shuffle. And this would be what this one would look like. So this is only happening in meiosis. And again, the whole point of this is just to create what these are now, these are what we would call these recombinant. So it's to create recombinant chromosomes. Eventually these will split up and we'll call them chromosomes. It's to create recombinant chromosomes so that when fertilization occurs, the offspring is sort of a genetic blend between mom and dad. And that may help the offspring and it may suck for the offspring, but it's just a roll of the dice. Are genetics, what we did in class today, going to be on the test? There will be some bonus questions involving Punnett squares and crosses, yes. I don't really get how chapter 50 related to our unit. Well, chapter 50 was all about, or at least the part that we talked about, it was all about cell communication. So this right here is a perfect example of cell communication, which is all chapter 11. We also talked about what happens when this process goes wrong. Like, for example, the sarin toxin interferes with this process and it will kill you. And botulism, or the botulism toxin interferes with this process and you will die. It also relates to what we've been talking about just in general in this class, because this is looking at protein form and function, and we're looking at how protein form fits function, and also how the shape of a protein is critical in its ability to do its job. And then finally, we also talked about a secondary messenger in this chapter, and that secondary messenger was calcium, and how important calcium's role is in enabling muscle contraction to occur by binding to troponin. So that's why I decided to introduce chapter 50 for this unit. What is CDK and MPF? Oh, hang on. What is an effector cell? It's a short-lived cell that is created when a lymphocyte is activated. So an effector cell, I don't know what we call effector cells for T cells. I know that for B cells, we call the effector cells plasma cells. You can also refer to it as an effector B cell. You're saying the same thing. So they're short-lived, they last for a few days, they do their job, and then they die. And they're different because memory cells really don't do anything except activate more rapidly when they're exposed to antigen again. So they're really important, but they're not actually responsible for going out and hunting down the pathogen. They're responsible for just remembering what it looked like. What is CDK and MPF? So CDK and MPF, MPF is when you take cyclin-dependent kinase, which is just a protein in the cytoplasm. It's a kinase, and as the name implies, it needs cyclin to do its job. And cyclin is a protein that begins to accumulate as the cell passes its checkpoints during interphase. So as the cell passes its checkpoints during interphase, it will produce more cyclin, and this cyclin will bind to CDK. And we call this complex of cyclin plus CDK, we just call it MPF. And when we see a high concentration of MPF inside of the cell, we know that it's about to do mitosis. So in other words, MPF just activates cell signaling pathways that ultimately result in the cell doing mitosis. Chapter 50 was about ecology. Indeed, chapter 50 in your hard copy is muscle contraction, I think. And then in the electronic text, muscle contraction is chapter 49. Can you discuss protein phosphorylation and tyrosine kinases? Sure. So a tyrosine kinase is a transmembrane protein that has a receptor region out here, a transmembrane region right here, and then finally a kinase region. And the specific amino acids that are actually getting phosphorylated are tyrosines. They could be something different, but the main one that we focused on is tyrosine. So that's why we're going to call this RTK. So when RTKs come together, like let's say this is a ligand that stabilizes the active form of the RTK, because RTKs can be like all scrunched up when they're in their inactive form, so they can't come together if they're all scrunched up, but binding to the ligand will stabilize its active form so that two can bump into each other and form a dimer. And when they bump into each other, they will phosphorylate each other. So this one will phosphorylate. The left will phosphorylate the right, and the right will phosphorylate the left. And this phosphorylation can activate multiple cell pathways. We focused on RTK as mostly activating growth pathways, but RTK also is used for the insulin signaling pathway. What is the difference between a phagocyte and a white blood cell? So a white blood cell, you can look at the Choose Your Own Adventure. There was a case study that talked about chronic myeloid lymphoma, or leukemia, sorry. But in chronic myeloid leukemia, in that case study, they started with a stem cell and they talked about all of the different cell types that this stem cell can transform into. And so one of the types of cells, or it's like a family of cells that this stem cell can transform into, are white blood cells. So within the white blood cell family, you have phagocytes, you have cells called granulocytes, you have cells called lymphocytes, you have cells called megakaryocytes. So there's a whole bunch of different types of white blood cells. So lymphocytes are a type of white blood cell, phagocytes are a type of white blood cell. And then you can even talk about types of phagocytes like macrophages and neutrophils and dendritic cells. So it's like this big family tree. No, you do not need to know about ecology for this test. Ooh, how do cells arise from zygotes and meiosis? So somebody's asking about how all of the different cell types arise from a single zygote. And that's a great question, and we'll cover that when we talk about gene expression and embryological development, but you don't need to know that for this test. So, yeah. Oh, there's a question up above. What happens when fertilization occurs and you have a zygote? Again, that's a really good question. So you have a little sperm cell, yeah, and a big egg cell, and these are both haploid. And when fertilization occurs, you have the formation of a zygote, which is now diploid. That zygote will undergo a bunch of rounds of mitosis. And as it undergoes mitosis, these cells will start talking to each other. And by talking to each other, they're figuring out what exactly they should transform into. So in a nutshell, there's the answer to that question, but obviously it's much more complicated. Stem cells are made in your bone marrow. Well, you have stem cells all over your body. The stem cell that we focused on for chapter 43 was the hematopoietic stem cell, which, yes, is located in your bone marrow. Uh-oh. I think my microphone was muted. I don't know how much I was saying with my microphone off. I don't know if you heard this, but stem cells are all over your body. We talked about the hematopoietic stem cells, which are located in your bone marrow. What does cyclic GMP do? Kind of does the same thing that cyclin does in this scenario. So cyclin binds to a kinase and activates it. Cyclic GMP binds to other kinases and activates those kinases. So it binds to kinases, changes the shape of the kinase so that it's active, and therefore helps propagate the signal through the phosphorylation cascade. There's a question about plants and protists and their life cycle phases. I didn't spend much time talking about it. Your book mentions it briefly, but I'm going to go back to it when we actually talk about protists, the alternation of generations, where basically you have one generation that is diploid and produces spores, depending on what species you're talking about. And then the next generation that comes from the spores is a multicellular haploid generation that will produce gametes, and those gametes will fuse to restore the diploid generation. But no, I don't think you're going to be held responsible for that on this unit exam. What is the difference between synapsis and chiasmata? Chiasmata is, you know what, I want to say that synapsis is the process of the homologous pairs binding to each other. So we didn't talk about the synaptomal complex, but basically these homologous pairs don't just like bump into each other and hope that they hit each other in just the right way so that crossing over happens. I mean, there's an entire mega structure of proteins that basically like staple these homologous pairs together and hold them together so that they can do crossing over. So synapsis is just where the homologous pairs are sort of like temporarily glued together. And then once they're glued together, then they can do crossing over. And then when they do crossing over, we can see like we can see them crisscrossing, and that's called the chiasmata. So I know they all sort of sound like the same thing. They're technically different. So synapsis is them coming together, and this would be like crossing over is the process. But then the actual structure where you see the crisscrossing, like right here, that would be called chiasmata. The G0 phase is where the cell is just doing its job, but it is not ever prepping for cell division. So all right, this will be the last one, the centrioles function. So the centrioles function is during mitosis or meiosis. Its function is to, at least in animal cells, plant cells don't have centrioles, so we really don't. There's still a lot of unknowns about what exactly the function of the centriole is, if you know what I mean. Because obviously if they're super, super important and plant cells do just fine without them, then maybe they're not so important. But as far as I understand it, the role of the centrioles in animal cells is to basically mass produce microtubules. And so you can think of a centriole as basically like a microtubule factory. And so they mass produce these microtubules, and these microtubules are going to reach out and attach themselves to the centromere, which is where the chromatids come together. And then also they're going to sort of like stretch past the chromosomes and like interact with each other. So they'll interact with each other and they'll basically start pushing against each other. And by pushing against each other, the cell sort of elongates like this. And so this is what helps split the cell or push the cell out so that it can eventually perform cytokinesis. And then finally, clonal selection is just where you have your B cells are turned on, your B cells are activated. And when they get activated, like we were talking about over here, they undergo massive rounds of mitosis and produce a bunch of plasma cells that each secrete antibody. All right, that's it. I'll see you guys tomorrow. Happy studying.